Hello and welcome to this Carvin Academy tutorial on A-level biology. In this video we will look at the process of mitosis, answering what it is, why it's important, and what happens when it goes wrong. So let's get started. So what is mitosis? Well, mitosis is a production of two genetically identical cells from one parent cell. We say that these cells are diploid, which means that they contain one chromosome from dad and one from mom. Mitosis is considered a type of asexual reproduction, where the cells made are called clones. They contain the same genetic material, a bit like the stormtroopers. I only added this to make this slide more appealing, otherwise it is completely unnecessary for what we'll learn today. Apart from mitosis, there are other types of asexual reproduction, such as binary fission, which is carried out by bacteria, and budding, which most fungi and some plants use. Before we learn about the process of mitosis, we need to understand its importance. Why do cells carry out mitosis, seeing that it requires a lot of energy? One of the most important needs for mitosis is cell replacement. Cells die every minute and every day, and they need to be replaced. Another need is tissue repair. It's not surprising that when we get injured, our body covers up the scars, and this is only possible through mitosis. Finally, mitosis is important for the overall growth of the organism. That's why you're not the same height or weight you were 10 years ago. And all this leads to survival of an organism. So mitosis is part of cell division. It's controlled mainly by two types of genes. The first type is called proto-oncogenes, also known as the accelerators, and activation of these genes cause cell division to occur. Note the prefix onco means tumor or cancer, hence why cancer doctors are referred as oncologists. The second type of genes are called tumor suppressor, also known as the brakes. Activation of these genes stops cell division. Thus, a balance between a tumor suppressor and a proto-oncogene is needed to reduce the chance of cancer or malignancy. However, mutations can occur in them. The proto-oncogenes become oncogenes when they mutate, and this causes rapid and uncontrolled cell proliferation, which leads to tumor formation. This diagram here illustrates what happens when proto-oncogenes mutate. The car accelerates too much with less brakes, leading to cancer. Mutation in tumor suppressors is also problematic because they can inactivate them. The inactivation of tumor suppressor means that cell division isn't stopped, and the cell proliferates, that is, it divides rapidly and uncontrollably, causing tumor formation. The diagram below illustrates that when tumor suppressors are mutated, the car has no brakes, so can't be stopped, so leads to cancer. There are many processes that occur before mitosis which are highly regulated. And because of this, most cells stay in G0 or quiescent phase, where they do not replicate and do their normal jobs. This is useful because it limits the chance of mutations and reduces unnecessary energy expenditure. Our cells only divide where and when necessary, such as sites of injury. Mitosis only makes part of the cell division. Other parts include interphase and cytokinesis. Interphase consists of gap 1, synthesis, and gap 2 phase, and it prepares the cell for mitosis, with G1 involved in organelle synthesis, S phase involved in DNA replication, and G2 phase involved in mitotic protein synthesis. So now let's get on to the phases that make up mitosis. There are 4 phases, which can be better remembered as PMAT. These stand for prophase, metaphase, anaphase, and telophase. So let's look at prophase. The prefix pro means first, hence why it's the first phase of mitosis. Once DNA has replicated, it exists as chromatids. Now it's important to understand the difference between chromatids and chromosomes. We have 46 homologous chromosomes during prophase, but there are 92 sister chromatids, because the DNA has replicated. I hope this image makes better sense. Note that sister chromatids are attached by a centromere, And also note that the homologous chromosomes mean that one chromosome is received from mom and one from dad, and these chromosomes have the same genes but different alleles. So now let's look at the process of prophase. During this phase, the chromatin, which is a looser form of DNA, condenses into chromosomes. It also involves coiling around histone proteins, which we'll learn about in a later video. Next, the nuclear envelope, which is a membrane surrounding the nucleus, breaks, releasing chromosomes in the cytoplasm. Next comes metaphase, and in this phase the chromatids move to the equator or the middle of the cell. These spindle fibers arise from the centrioles and they attach to the centromeres of the chromatids. The spindle fibers are also known as microtubules, and these terms are synonymous. After metaphase is anaphase, and in this phase the centromere divides, that is, it breaks apart. Then the sister chromatids move to opposite poles, that is, opposite sides of the cell. Each pole gets equal amount and identical copies of chromosomes. Finally comes telophase. Telo means last or final, hence why it's the last phase of mitosis. In this phase, the chromosomes decondense into chromatin and the nuclear envelope starts forming. After mitosis is finished, cytokinesis occurs. Note that cytokinesis is not part of mitosis. During this, the cells separate to form two identical diploid daughter cells, and this happens as a new cell membrane starts forming. So that finishes mitosis, and let's summarize what we learned in a slide. So cell division starts with interphase, which consists of G1, S, and G2 phases, and these phases all prepare the cell for mitosis. Mitosis is a production of genetically identical diploid daughter cells, and it consists of prophase, metaphase, anaphase, and telophase. Mitosis is needed for cell replacement, tissue repair, and growth. Proto-oncogenes and tumor suppressor genes activate and stop cell division respectively, and mutations in these genes can lead to rapid, uncontrolled cell division, which can lead to cancer. And that is mitosis in a slide. I hope you learned something from this video, and if you did, please make sure to like and subscribe. Thank you, and I'll see you in the next one.